In my seminar today, I will talk about the chemical complexity that can be attained in the interstellar medium and about its possible contribution for triggering prebiotic chemistry. So before starting, I would like to give some context. I'm an astrophysicist working in the Centro de Astrobiología, CAP, in Madrid. At CAP, I am surrounded by colleagues from very different fields: biologists, chemists, geologists, engineers, and planetary scientists. Researchers from very different fields that are trying to understand from different perspectives how life could be originated on Earth and whether it's possible somewhere else in the universe. In our case, from the interstellar medium group, what we want to understand is how complex is the chemistry that can occur in space in the diverse sites of stars and planets, and whether it could contribute to trigger prebiotic chemistry on early Earth, and whether it has the potential to do it in other planets. So this is a simplified timeline of events pertaining to the early history of life on Earth. When we try to understand how life appeared on our planet, there are two kinds of approaches. On one side, top-down approaches, which include phylogenetic trees or fossil records that tell us that life appeared at least around 4 gigayears ago. These kind of studies face, however, some limitations, because the farther back in time we go, the less clear the evidence becomes. So at the end, we face a sort of informational horizon. As a result, we lack evidence of the conditions on early Earth, including proof of the molecules that might have formed the building blocks of life. So complementarily, alternative bottom-up approaches have been also explored, which try to understand what could happen from the other side of this informational horizon since the formation of the Earth around 4.5 gigayears ago, like prebiotic chemistry on Earth, or even before that, interstellar chemistry within the NATO molecular clouds. So I am an astrophysicist, so please let me give you a very basic picture about how and where stars and planets form. Their clouds are interstellar clouds composed by dust grains and molecules, and if you observe a molecular cloud in the infrared, as in the left panel, it will appear as a dark silhouette because the visible and near-infrared light is blocked by the high amount of dust. However, if we observe them with larger wavelengths in the centimeter and submillimeter ranges, which are not absorbed, we realize that these clouds are indeed full of molecular material, like shown in the center and right panels, which show the emission of two abundant molecules in space, N2H+ and SiO. These nanomolecular clouds have typical densities of 10 to the 3, 10 to the 4 centimeters cubic, and temperatures between 10 and 20 Kelvin. And as I have mentioned, contain a great variety of molecules inside. In this environment, some denser condensations can appear, starting the process that will form stars and eventually planets around them, as you can see summarized in this diagram. All these different phases can be directly observed, and I show you here real observation of each one of them. Natural molecular clouds, seen as dark lanes in this infrared image, dense dusty cores formed by gravitational collapse that surrounds the burning protostars, protostars that ejects powerful outflows to conserve the angular momentum, and the dusty and molecular envelope is progressively cleaned up, and at the end, a protoplanetary disk will appear like the beautiful HL tau disk imaged by the radio interferometer ALMA I show you here. And as a final product, a planetary system will appear with the central star, planets orbiting around it, and also comets and asteroids. One of the main aims of astrochemistry is to follow this whole process using the emission of molecules to understand what is the chemical thread that links the different phases of formation of stars and planets, and what is the full chemical reservoir that a forming planet can receive. And this is not obvious because, in principle, the interstellar medium is a very hostile environment to allow chemical reaction because it is cold, its density is very low, and there are high levels of radiation. But astrochemical studies in the last decades, and especially in the last few years, have shown us that the interstellar medium is indeed full of molecules and that the chemical complexity revealed is relatively rich. We can indeed say that we are living a golden era for astrochemistry, and a very illustrative way to show this is these plots that show the cumulative number of molecules detected in the interstellar medium as a function of the year. From the lesion detections of the late 30s, there is a continuous increase on the number of molecules. We have today more than 300 molecules detected, with an impressive increase in the detection rate in the last few years. Since 2021, more than 80 molecules have been discovered, which means a real revolution in astrochemistry. I show you here a list, which is not fully updated, of the molecules identified so far in the interstellar medium. So from now on in the seminar, I will try to answer these 4 different questions. Among of the molecules that might be present in the interstellar medium, in which ones we are primarily interested and why. I will explain what strategies we use to search for them and where in space we search for them. And to conclude, I will show you some examples of our efforts to understand how the molecules can be synthesized in the interstellar medium. So let's start with the first question: which molecules are our prime targets? So we know that the origin of life required a process of continuous increase of molecular complexity, from relatively simple molecules to larger biomolecules. So the question is, what simple chemical compounds could be realistically available at the dawn of life, and what was its origin? One possible option is the endogenous synthesis, in which the organic compounds might be produced from very simple molecules through atmospheric reactions on early Earth. An alternative could be the exogenous delivery, namely that organic precursors arrive to the primitive Earth transported by extraterrestrial objects. This hypothesis is supported by the detection of relatively complex molecules relevant for life in meteoritic or cometary material. I show you here the recent detection of uracil, RNA nucleobase, in the samples of the asteroid Ryugu that have been transported into Earth by the Japanese Hayabusa 2 spacecraft to perform careful analysis. This discovery provides further evidence that the important building blocks for life are created in space and could have been delivered to Earth by meteorites or comets. Other works, in this case in the laboratory, have also proposed ways of forming peptides, which are protein precursors at the low temperatures present in the interstellar medium, starting from very simple molecular precursors that are abundant in space. Therefore, the exogenous delivery appears as a plausible option to provide at least a fraction of the chemical feedstock that could have triggered prebiotic chemistry on early Earth. And especially because we know that around 3.9 gigayears, our planet suffered a heavy bombardment of meteoritic and cometary material. And this time was indeed very timely, since this happened just before life appeared. So it is reasonable to think that the input of extraterrestrial material could play an important role for the development of prebiotic chemistry. It is then very relevant to understand what kind of molecules can be formed in the interstellar medium, allowing its incorporation to the material that eventually will form planetesimals. So the basic questions we aim to address is whether the molecular precursors of life are present in space. And if this is true, the implications will be deep, since it will imply the universality of the basic inputs for life, and this means that life might not be a unique property of our solar systems, but also of other sites in the universe. So to guide our search of molecules with prebiotic interest, and following the advice of our colleagues here in the Centro de Astrobiología in Madrid, we have followed an approach based on the RNA world, which is deemed the most plausible framework for the origin of life. Very interestingly, a series of laboratory experiments in the context of the RNA world carried out in different research groups have shown that RNA nucleotides, here on the right, can be formed from quite simple molecular precursors on the left. And among them, we have well-known interstellar molecules, species that have been already identified in the interstellar medium. Obviously, the full pictures can be much more complicated, but the message behind is the same. From simple molecular precursors, we can develop the chemistry needed for the RNA world. Therefore, these simple precursors are the molecules that we want to search for in the interstellar medium. So several years ago, in our group at the Centro de Astrobiología, we designed a roadmap based on the outcome of prebiotic experiments in the lab to organize the query of key prebiotic precursors in space. I show you here our first simple scheme in which you see simple precursors on the left and prebiotic species on the right, including ribonucleotides. Molecules already detected in space are indicated with solid boxes, and species still to be detected are highlighted with the dashed boxes. These were the prime targets of our search. So how can we afford the detection of these molecules in the interstellar medium? The molecules in the gas phase of the interstellar medium exhibit different kinds of motions, including bending, stretching, and rotations. And these motions produce emission of photons at very particular frequencies in the centimeter and submillimeter range in most cases that we can measure with our telescopes. I show you here a few examples of the facility I often use, like ALMA, the Atacama Large Millimeter Array, which is composed by more than 50 antennas in the Atacama Desert in Chile, or large single-dish antennas such as the Iran 30-meter in Pico Veleta in the south of Spain, or the 40-meter antenna at Jeves close to Madrid, also in Spain. Obviously, as you can imagine, the rotational spectrum of the molecule, which is the fingerprint of the molecule, is crucial information that we need to know before trying to identify a molecule in space. These spectra can be measured in the laboratory by our colleagues, spectroscopists, which provide us the spectrum of the different molecules that we can compare with the spectrum observed pointing our radio telescope towards a position in the sky. And I want to stress that this synergy with the laboratory is absolutely essential, especially when we search for molecules with prebiotic relevance, because for them, often there is no spectroscopy available, which prevent its search in space. So, okay, once we know what species we want to detect, an important question is where we should look at to detect them. This is a diagram of the formation of stars similar to the one I showed at the beginning. We are interested in understanding the chemical feedstock during the different phases of star and planet formation, and to know if there is any hint of chemical heritage during the process. There is indeed evidence pointing that the chemistry during these different phases shares some similarities. For instance, looking at the molecular abundance ratio between methyl formate and dimethyl ether, the abundance is close to 1 and very similar regardless of the type of source, from high-mass protostars, low-mass protostars, protostellar shocks, prestellar cores, galactic center molecular cloud, and also in comets. And when comparing the chemical composition of star forming regions, we represent initial phases of star formation, and that of the comet 67P, Churyumov-Gerasimenko, will represent the leftovers of the formation of our own solar system. It has been seen that they are reasonably similar. Drobdovskaya and Álvaro López Gallifa, a PhD candidate in our group, have shown that the molecular abundances of a low-mass protostar on the left and a high-mass protostellar cluster on the right show a good correlation with the chemical content of the comet 67P. And this suggests that the bulk of the chemical reservoir is preserved during the star formation process. So let's start the query of new molecular species highlighted as key precursors in prebiotic experiments. We started looking to a star forming region, and we draw our attention to a species that was a key precursor of both lipids and ribonucleotides, according to the work by Patel 2015. And this is glyconitrile. So we pointed our telescope, in this case the ALMA observatory, towards the region IRA 16293, which is a prototypical protostellar system with a rich chemistry. This image shows the dust emission surrounding several protostars in the system. So we detected successfully glyconitrile for the first time in the interstellar medium towards one of the protostars of this protostellar system. This means that the gas that is surrounding the protostar is rich in a key RNA precursor. So coming back to our starting roadmap, we were able to provide the detection of one of the key species that was still elusive in space. But of course, we did not want to stop here, and we aimed to detect many more new species. And in order to do that, we moved our attention to a previous step in the star formation process, the nasal molecular clouds. In particular, to a molecular cloud located in the center of our galaxy. I show you here a beautiful image of the center of the Milky Way, and in the last years, our group at CAP has been devoted to perform deep searches of new molecules towards the molecular cloud G0693, which is located in the Sagittarius B2 region of the center of our galaxy. So before showing our latest results, let me briefly motivate why we choose this particular region. It was first studied back in 2006 when several oxygen molecules were detected, like methanol, ethanol, dimethyl ether, or methylformate. 2 years later, more complex species with oxygen, such as propanol, ethylene oxide, or ethylene glycol, were also detected. But the cloud is also rich in molecules of other chemical elements, such as nitrogen and also phosphorus. So at that point, we wondered if this source might be the right place to address the detection of molecules not seen before in space, namely, if this source could provide also first detections in the interstellar medium, and in particular of species with prebiotic interest. And this led us in 2019 to the first detection in space of the Z isomer of cyanomethanamine, which has been proposed as a possible precursor of adenine. This detection confirmed us that G0693 was indeed a very suited laboratory to detect new species. But why this source exhibits such a rich chemistry? So this cloud is located in Sagittarius B2 complex in the galactic center, which harbors an active protostellar cluster of massive stars with two main sites called N and M that are clearly visible in this ALMA image. Our molecular cloud is located 1 arc-minute towards the northeast, and as you can see, it seems that there is nothing there. But this is not true at all, and indeed, the cloud is full of molecules. Thanks to interferometric maps, we knew that the region is very rich in SiO emission, which is a well-known shock tracer. This SiO emission is due to the presence of large shocks in the region, likely from a cloud-cloud collision. And this has important consequences for the chemistry, because the spattering of the grains produced by these shocks are able to release into the gas phase the molecule formed on the grains, producing a very rich chemical content in the gas phase. Indeed, when we map the emission of a complex species, such as ethanol, with the 830-meter telescope, it was clear that the peak of the molecular emission is precisely centered at the position of G0693. So therefore, this molecular cloud is a well-suited candidate for the detection of new species. So for this reason, we have been conducting in the last years an unbiased and very sensitive spectral survey of this cloud using different facilities, as you can see here, such as the GBT, the JEVES 40-meter telescope, the 830-meter telescope, and also APEX, covering frequencies from 12 to 275 gigahertz with a total coverage of almost 200 gigahertz. I show you here in the different panels the spectra of the survey in black, along with the fit of all the molecules we have identified so far. And as you can see, the amount of detected lines is impressive. So far, we have identified more than 130 molecules toward this source, including many first detections in space. So let's start with some of the prebiotic species that we have detected toward this source in the last few years. One of them is hydroxylamine NH2OH. We were very interested in this species in particular because many prebiotic experiments propose that it is a key prebiotic precursor to form RNA nucleotides, while several other precursors were already detected in the interstellar medium, like formaldehyde, hydrogen cyanide, cyanoacetylene, cyanamide, or glycolaldehyde. Hydroxylamine was elusive. And its non-detection was quite surprising for astrochemists because it is a relatively simple molecule with only 5 atoms, and chemical models predicted that this should be rather abundant. So we decided to search for this molecule using our deep spectral survey towards G0693 molecular cloud, and we were able to identify for the first time in space this species, thanks to the detection of several of its rotational transitions, as you can see here on the right panel. So hydroxylamine was the first species detected in space with an amine and an hydroxyl group. So we wonder, can we detect other similar species with increasing complexity? And in particular, we focus our attention on this one, ethanolamine. And we were very interested on this molecule since within the RNA world, there is another agent that for sure played a crucial role, which is compartmentalization, namely the presence of membranes. Membranes, as you know, encapsulate and protect the genetic material and enable the metabolic activities within the cell. And the membranes of all correct cells are made of a lipid bilayer of phospholipids, which are composed of a polar hydrophilic head and 2 nonpolar hydrophobic tails. I show you here in the middle phosphatidyl ethanolamine, which is the second most abundant phospholipid in living organisms. And if we take a closer look to the head, we see that the nitrogen-bearing head group is ethanolamine, a molecule that we thought that was potentially detectable in the interstellar medium. The species was already being identified in the Alma-Ata meteorite, which fell in the desert of Sudan some years ago. However, the origin of ethanolamine was not clear, and 2 options were proposed: in situ thermal decomposition of amino acids under a specific unusual condition of the parent asteroid, or alternatively formation from smaller interstellar precursors prior to the formation of the asteroid. Then, encouraged also by this detection in a meteorite, we decided to search for ethanolamine in G0693. And the exquisite noise of the spectra allows us to clearly identify at least 14 transitions of these molecules that are not blended with other species, which are shown in this plot. The detection of ethanolamine in the interstellar medium has interesting implications for prebiotic chemistry, because it is clear that to understand the origin of life, one should also understand first the origin and early evolution of cell membranes. And it is thought that the very first cells were composed by relatively simple fatty acids and alcohols. And the availability of ethanolamine in the interstellar medium and the detection in meteoritic material suggests that this molecule could have been delivered to the early Earth through meteoritic impacts. This could have been enabled the progressive replacement of fatty acids and alcohols by more efficient and robust molecules such as phospholipids, playing a key role in the early evolution of cell membranes. And indeed, laboratory prebiotic experiments have proposed that phospholipids can be formed using ethanolamine as a precursor under plausible earlier conditions. Moreover, given its similar chemical structure, ethanolamine has also been proposed as a direct precursor of the simplest amino acid, glycine, in simulated hydrothermal vents. The next molecule I want to show you today is 1,2-ethanediol. We were interested in this species because this has also proposed as an important intermediate in a prebiotic experiment. So coming back to this complex diagram, if we zoom into this region of the organic precursors, we have this reaction sequence here, which starts with glycolaldehyde. According to prebiotic experiments, glycolaldehyde can form this other species, 1,2-ethanediol, which is an isomer of glycolaldehyde indeed, which acts as intermediate to form the 3-carbon sugar glyceraldehyde. But the sequence does not stop here, but can continue forming the hydroxyacetone and a final product that is one of the big fish of prebiotic chemistry, glycerol, which is other of the building blocks of phospholipids. So in this sequence here, only the very first step, glycolaldehyde, was detected so far in the interstellar medium. So we wondered, what about the second species in the sequence, 1,2-ethanediol? However, we had a serious problem here. We were not able to search for this species in the interstellar medium because there was no rotational spectroscopy studied in the lab. So that's why I asked my spectroscopy colleagues, Mattia Meloso and Luca Bizzocchi, about the possibility of measuring the rotational spectra of these molecules in their lab in the University of Bologna. And they started to work on that. It was not an easy task, but at the end, they found a very clever way to produce the molecule directly in the lab using a pyrolysis technique and measured the spectra. So I took the freshly obtained rotational spectroscopy they provide me and searched in G0693 spectral survey, and the molecule was also there. So a new key precursor of our initial roadmap detected in space. So let's move now to another related topic in which I work actively in the last years, molecule with phosphorus. I've been very interested on the search of molecule with phosphorus in interstellar medium because, as all of you know for sure, phosphorus-bearing species, in particular phosphates, are absolutely crucial for biology. Their good chemical reactivity and at the same time structural stability makes them perfect to build up macromolecules like ATP, phospholipids, RNA, or DNA. Moreover, prebiotic experiments have shown that phosphorus is a key catalyst and chemical buffer for the formation of RNA nucleotides. So back in 2016, we detected for the first time PO in a star-forming region, which is the very basic building block of phosphates towards a couple of star-forming regions. And later in 2020, we also detected PO for the first time in a comet this time, using the amazing data provided by the Rosetta mission of the comet 67P Churyumov-Gerasimenko. So we learned that PO is the main carrier of phosphorus in the comet, and we know that this is also the case in the star-forming regions, based on previous works by our group and other groups. That's fine. So, perfect. So I can continue from here. So this is about the discovery of PO in the comet 67P. So what we learned is that PO, like all the volatiles detected by Rosetta, was already in the cometary ices prior to the birth of the sun, and has this indicated that the chemical budget of the comet was inherited from the natal environment of the solar system. So encouraged by the relevance of phosphorus for prebiotic chemistry, we also searched for phosphorus-bearing species toward G0693. And as expected, we detected PO and PN, which were detected previously in several star-forming regions. But in this case, we wanted to make a step further, and we wondered if we were able to detect new species with phosphorus not seen before. And since NO plus and SO plus cations are detected toward the cloud, we thought that maybe PO plus might be also there. And we were lucky again, and thanks to the extreme sensitivity of our spectral survey, we were able to detect two rotational transitions of the molecule, confirming its presence in the interstellar medium. So I hope that I have already convinced you that G0693 molecular cloud is an astrochemical mine with a very rich chemistry. Coming back to this timeline, in 2020, we detected hydroxylamine, already mentioned, but also propergylamine, just after obtaining its spectroscopy in the lab by Luca Bizzocchi at Paolo Scaselli's group at MPE. 2021 was a particularly prolific year in which, besides ethylamine, we also delivered the first detection of other species such as thioformic acid, ethylisocyanate, ethylmercaptan, thianomidyl radical, and vinylamine. And in 2022, we published the detection of ethanediol and PO+, I already mentioned, but also of two conformers of N-propanol in a work led by Izaskun Jimenez Serra. And early last year, in collaboration with David Alberton at NP, we measured the rotational spectra and provided, in this case, a tentative identification of 2 isomers of tralsalidimine. And moreover, in collaboration with Mariam Fatima and Holger Müller in the University of Cologne, who measured the spectra in the lab, we reported the first detection in space of isobutene. But the story does not end here. Indeed, we have now significantly deeper data of G0693 thanks to new recent observations. So we have observed with the Jebes telescope in the Q band from 31 to 50 gigahertz during more than 230 hours, reaching a wonderful sensitivity of less than 1 millikelvin, which improves the previous one by a factor up to 5. Moreover, we have also recently obtained new deep Eton 30-meter observations to improve the noise of other spectral windows, reaching sensitivities around 1 millikelvin as well. And this will give access to a new catalog of molecules whose weak lines were hidden in the noise so far. So let me come back a second to this life timeline, because we know that during the RNA war period, it is thought that proteins already appear, allowing the transitions to the subsequent DNA proteins world. And as a consequence, given the key relevance of proteins for biochemistry, one of the most desired items for astrochemistry would be the detection in the interstellar medium of the simplest amino acid, glycine. Several chemical models and laboratory experiments indeed have demonstrated that in principle, glycine should be able to be formed under interstellar conditions. However, although has been detected in comets, not yet in the interstellar medium, despite numerous observational efforts. So glycine is a C2H5O2N isomer, but it's not the only one. I show you here in this table other glycine isomers, orders in terms of thermodynamic stability, from low to high energy. So what we have done is to use our brand new ultra-deep spectral survey toward G0693 to search for all these different isomers. Indeed, not all of them, because unfortunately there is no rotational spectroscopy available for all. We have searched the ones for which spectroscopy is available, indicated here in blue. I show you here the 3D representation of these isomers. And I'm happy to say that this search delivered the first detection of a glycine isomer in the interstellar medium. We have detected several unblended lines of the lower energy isomer of glycolamide. This is species you can see here. And as you can see on the figure here on the left. And as I mentioned, this is the first isomer of glycine ever detected in the interstellar medium, and also the largest molecule containing the amide functional group that is unambiguously detected in space. So glycolamide, this species here, is another example of molecule containing the NH2 and the OH groups, along with the hydroxylamine and ethylamine I presented before. So the other glycolamide isomers, including the 2 conformants of glycine, are not detected, and we have obtained very low upper limits for their abundances. For the case of glycine, the upper limit is very similar to the abundance derived for glycolamide, which indicates that glycine is likely less abundant. But one can wonder if this is so surprising. In other words, should we expect the detection of glycine? And glycine is a carboxylic acid, molecules with the chemical group COOH. These species are widespread in nature and, as you know, are precursors of biologically relevant molecules such as amino acids. However, they have a problem for the astrochemical point of view, and because they are pretty scarce in the interstellar medium. Indeed, only 2 of them have been detected so far. Formic acid, more than 50 years ago, and acetic acid 25 years ago. So the census of carboxylic acids has remained the same for almost a quarter of a century, despite the boom of interstellar detection in the last decade. So they should make us think that the detection of more complex carboxylic acid is going to be really challenging, and maybe glycine is not the easy one to try. It includes also another carbon and nitrogen atom. So what about other candidates that might be more easily detectable, considering that only formic and acetic acid has been detected so far? So we thought that carbonic acid was a very good candidate given the high number of interstellar molecules detected so far with the OH group. And happily, our intuition was not wrong at all, since we have recently detected carbonic acid for the first time in space in a work led by a postdoc in our group, Miguel Sanz Novo. This species is not only the third carboxylic acid detected in interstellar medium, but also the first molecule with 3 oxygen atoms detected in space. And this is highly interesting because on one side opens the window for the detection of more carboxylic acids, including, of course, the long search glycine. And on the other side, give us also new hope for the detection of a species with 3 oxygen atoms of high prebiotic relevance. So coming back to the detection timeline of G0693, we have recently added these other 2 species, glycolamide and carbonic acid. And now I'm in trouble because I have no more space in the slide. And of course, this timeline would stop here. And early this year, Miguel Sanz Novo has published the new detections of the cation HOCS plus and thionyl imide HNSO, which is also highly interesting because it's the first interstellar molecule that contains simultaneously nitrogen, sulfur, and oxygen. And this implies a direct chemical link between these 3 elements key for a living organism. Moreover, David San Andres de Pedro, a PhD candidate in our group, has recently detected for the first time in space the N-isomer of cyanomethanamine, H2CN-CN, which is an HCN dimer. Therefore, our work has revealed that G0693 molecular cloud is an astrochemical mine with 18 new interstellar molecules discovered in the last 5 years. And we want to stop here, so stay tuned in the following months. Okay, at this point, one may wonder what happened beyond our galaxy. Are also prebiotic precursors in the gas of other galaxies? And the best dataset to answer in detail this question is a deep and unbiased spectral survey that has been done as an ALMA large program, Alchemy. We have observed, using a very broad bandwidth available at ALMA, the starboard galaxy NGC 253. We have identified tens of different molecules, including relatively complex species, such as ethanol, and we also search for species with a prebiotic interest, in particular simple species with phosphorus. And in a work of another PhD candidate of our group, David Hasler, who was indeed an undergraduate at that time, we provided the first detection of a phosphorus-bearing molecule, PN, outside our own galaxy. So in the last minutes of the seminar, I will discuss about how we are trying to understand how these molecules are formed in the interstellar medium. Generally speaking, there are 2 main ways of forming molecules in the interstellar medium. Directly in the gas phase, through the process indicated here, such as ionization, dissociation, and 2-body chemical reactions, such as, for instance, ion-neutral reactions. And alternatively, molecules can also be formed on the surface of interstellar dust grains, which can offer some advantages that makes the chemical reactions easier and more effective. I want to stress that we know that both ways are known to work, depending on the physical conditions and also depending on the molecule, and that they are fully complementary. So this yellow square is, believe me, is the foundations of current astrochemistry. So in order to fully understand how the chemical complexity in space can arise, we absolutely need the interplay of different fields, because astrochemistry is intrinsically interdisciplinary. In one of the corners of this box, we have the astrochemical observations that I have already presented in the seminar. I already mentioned that to search for a species and to derive properly the molecular abundances, we need the spectroscopy measured in the lab. But the labs can also be extremely useful to mimic the physical conditions of the interstellar medium, and they can suggest us possible ways to form the molecule, which is of great help when we try to interpret the observations. But the experiments cannot do everything, and in some cases we can just model the chemistry. So we use complex chemical networks coupled with physical models of the interstellar sources, which are fed by the outcome of observation and experiments. This model can tell us which chemical pathways are relevant and thus how the molecules we detect are formed. However, the chemical networks are based often on incomplete or directly unavailable data, which is simply assumed. So to fill this gap, we need to perform quantum chemical calculations that address the kinetics and thermodynamics of the chemical systems and provide us useful information about the chemical reactions such as exothermicity or reaction rates, which are then introduced into the chemical models to provide more accurate predictions to be compared with observations. So only the continuous synergy between these 4 different fields will allow us to understand better how interstellar chemistry works. And in the last slides, I will briefly show several efforts we have done recently to understand how the molecules that we have detected for the first time in space can be formed in the interstellar medium. So in the case of hydroxylamine, previous to our detection, it was proposed by laboratory experiments that it can directly form by sequential hydrogenation on the surface of dust grains, starting from NO, which is an abundant species. We know now that this route is likely the main contributor to the formation of hydroxylamine. However, there was a serious discrepancy of several orders of magnitude between the abundance predicted by the models 10 to the minus 6 with that observed 10 to the minus 10. So with the motivation of solving this problem, I asked my collaborator Germán Molpeceres, which is an expert on quantum theoretical calculations, if he could explore the extraction mechanism of this molecule that could better match the low abundance derived in the observation. And in a work published last year, he found that there are 2 main possible ways of destroying efficiently these species on the surface of grains. One, working on 10 Kelvin, which is the possible hydrogenation of NHNO that can follow the sequence that will end with hydroxylamine, but can also form NO plus O2, NO plus H2 molecular hydrogen. Germán found that enabling 15–30% of this reaction results in a reduction of 1–2 orders of magnitude of the hydroxylamine abundance. Moreover, he also studied theoretically in detail the possible destruction of hydroxylamine by combination with atomic hydrogen and found that the reaction at 20 Kelvin is efficient enough to account for an important destruction of the molecule that will decrease the abundance to the levels observed. So this work has highlighted the importance of consider both formation but also destruction mechanism, which are usually overlooked to account for the molecular abundances derived in the observations. In other work, also in collaboration with Germán, we have proposed a new general pathway to form complex organic molecules on the surface of dust grains. Our approach starts with closed-shell hydrocarbons, acetylene and ethylene, which are thought to be abundant on grains, and study its reactivity with radicals on that surface. In particular, we studied the interaction with atomic hydrogen, OH, NH2, and CH3. And Germán calculated theoretically the possible reaction that takes place using this species. And as you can see, the same chemical reaction, C2H2 plus hydrogen, can give different products. However, we found that only some of them are exothermic and hence possible in the interstellar medium. So knowing that, the next step is to calculate the reaction rates to know if the reaction is indeed efficient enough. I saw here the reaction rate of the exothermic reaction of C2H2 with the different radicals we have explored as a function of temperature. And as you can see, only the reactions with H and OH have high reaction rates. So we expect that only the reactions can take place efficiently on the surface of grains. These reactions can form the radicals C3H3 and C2H2OH, which are highly reactive, and can then combine with other radicals available on the grains to form more complex species through radical-radical reactions. So this general mechanism is able to form many of the molecules detected so far in the interstellar medium, including some of the molecules that our group has detected for the first time in space. For instance, ethanolamine. Since we discovered this species for the first time in space, not much was proposed in the literature for it. I only found a possible chemical pathway proposed by Steve Charley, which is based on the hydrogenation sequence on the surface of that grain, which is indicated here. The different intermediates in the reaction may be directly formed through different reactions indicated in the diagram, which are based on different laboratory and theoretical works. This diagram summarizes all the possible routes that we thought that were reasonable when we published our detection of ethanolamine. However, the general theoretical work I have just shown about the surface chemistry done with Germán is also able to form ethanolamine, started with ethylene on the surface of a dust grain. The combination with OH is exothermic, and the reaction rate is high enough to form the radical C2H2OH. This radical can further react with an NH2 radical on the surface and form ethanolamine. So this is another way to form this species in interstellar medium. In any case, all these possible pathways proposed need to be well characterized, both theoretically and actually experimentally in the lab, and then introduced in chemical models. Will tell us which mechanism is indeed dominant. So I'm running out of time, so I will skip this and go directly to the formation of PO. In this case, what we did was to include this newly detected species into the phosphorus chemical network already developed by Izaskun Jimenez Serra and Serena Vitti, and couple it with a physical model that reproduces a shock with the aim of mimicking what is happening in G0613. So I show you here the prediction of the model as a function of time of the shock with different assumed values of the cosmic ray ionization rate in the cloud, which is an important parameter for the chemistry. Different solid color lines indicate the expected abundance of phosphorus beryllium molecules, and the dashed horizontal lines are the values derived from observations for PO and PO+. We have concluded comparison of observation and the results of the model that to explain the observed abundance of PO+, you need two basic ingredients. On one side, enhancement of phosphorus abundance in the gas phase, thanks to the presence of the shock, and high values of the cosmic ray ionization rates. So on one hand, the shock spatters the easy gray mantles, releasing into the gas phase most of their phosphorus content, mainly in the 4-os-phosphine in the model, which is then converted to atomic phosphorus, which is ionized efficiently by cosmic rays, forming P+. Later, further gas phase reactions with O2 and O produce PO+. Additionally, there is another route, which is the cosmic ray ionization of PO, that might also contribute significantly to the formation of PO+, and which will help to explain the quite high PO+-PO ratio observed in the cloud. So rather than conclusions, I want to conclude here with some takeaway messages of this seminar. First, the interstellar chemistry is able to synthesize relatively complex molecules in the interstellar medium. The availability of the spectroscopy of the molecule is absolutely key to search for new species in space. We have seen that the deeper the observations, the more species we find, and this is clearly indicating that the observed complexity is still limited by the sensitivity of the instrumentation of current telescopes. We need new theoretical calculations, models, and laboratory experiments to understand how the molecules that we are detecting are formed in an interstellar medium. And many of the basic chemical ingredients of the RNA world are available in the interstellar medium, which opens the possibility of triggering prebiotic chemistry and eventually life in other places in the galaxy under the proper environmental conditions. So thanks you very much for your attention. I will be very pleased to answer questions.